Yeah, great. So I want to thank Manish and Dr. Bailey for the invitation to come speak to you today. It's my first time out in Madison, so it's been really nice. I got a nice tour of the campus this morning. It's beautiful. So I'll begin with the disclosure slide. Unfortunately, I don't have anything that exciting to disclose. And I'll sort of set up the talk with the following overview. I'll talk a little bit about why are we studying ovarian cancer, what are the challenges, why does it make it interesting, talk a bit about how the pathology and understanding of the genetics over the last 5 to 10 years has really sort of driven our understanding of this cancer in ways that we didn't appreciate before, and then how that has sort of speared our approaches towards modeling this disease at the laboratory bench. So ovarian cancer, the challenges. If you look at the World Health Organization statistics, there are about almost 250,000 new cases of ovarian cancer diagnosed every year, with about 152,000 deaths from that disease per year. And unfortunately, it's because the vast majority of women are diagnosed at advanced clinical stage 3 and 4, where you get tumors that look like this. This is the actual tumor down here on the right. This is the uterus itself. And as many of us are familiar with the fact that you don't get symptoms until these tumors rise out of the pelvic brim, so to speak, and cause nonspecific GI symptoms. So there are no sort of hallmark symptomatologies associated with ovarian cancer. And unfortunately, we don't have any early detection for ovarian cancer either. And so this has made ovarian cancer the most lethal gynecologic cancer. And if you look at American Cancer Society statistics, when you look at the mortality to case ratio, ovarian cancer is actually third behind pancreas and lung. So it's actually much more lethal than many other cancers, even though the frequency of this disease is not as much. And you notice down here is breast cancer. Now, as a result of all these things, it's not surprising to find, if you look at statistics around the globe, that the 5-year mortality and survival rates for ovarian cancer have really not changed. You're looking at the US SEER database on the top here, data from Australia and from Canada, and it's pretty much been a flat line for a few decades. It's not really dramatically improved over time. This is further complicated by the fact that you're not dealing with a single disease. Ovarian cancer is a heterogeneous disease. There's multiple histological subtypes. And what this slide is intended to illustrate is the fact that the 4 major subtypes, serous, endometrioid, mucinous, and clear cell cancers, were all thought to arise from a single layer of cells that envelop the ovary called the ovarian surface epithelium. But not only do these tumors look different under the microscope, they all present clinically differently, they respond to therapy differently, they have their own unique natural history. And so about almost now, we're approaching 12 years or so, 13 years or so, Bob Kerman and Yimeng Shi at Hopkins decided to see if they could classify and categorize these tumors in ways that would make them more amenable to molecular diagnosis and hopefully translate that back to the clinic. And so they came up with their sort of now famous 2-pathway hypothesis for ovarian cancer. You have the type 1 tumors and the type 2 tumors. The type 1 tumors are the low-grade, slow-growing tumors. They comprise all the histological subtypes, and they're all listed here. They have mutations in all your favorite cancer genes, and the more that we've learned from the Cancer Genome Atlas and other studies, the more mutations we find in these tumor types, and oftentimes they're associated with precursors in the ovary, either endometriosis or some other precursor lesion. Type 2 tumors are starkly contrasted with type 1s in that they're always high grade and very aggressive clinically. The poster child for this one is serous, the high-grade serous carcinoma, and these are defined by mutations in p53. They also happen to be the subtype that you most often see with BRCA1, even though now there's emerging data that says that if you have BRCA1 mutations, you may get other subtypes of ovarian cancer as well, but this is the most common one. And until recently, there were really no known precursors. In fact, some people used to say that high-grade serous cancers arise de novo on the surface of the ovary with no pre-existing precursors or morphology to appreciate. So it was actually the cloning of the BRCA genes in the mid-90s that actually led to some insights into pathogenesis that I'll talk about. And so the BRCA1 and BRCA2 genes were cloned 1994, 1995, and shortly thereafter, by the end of that decade, we were offering women prophylactic surgery. So if you had a BRCA mutation, you could have your fallopian tubes and ovaries removed to reduce the risk of getting high-grade serous cancer. And this afforded pathologists the opportunity to look for occult malignancies. Right? In women who are thought to be completely healthy, no known evidence of disease, just a risk factor, were there any instances where you saw precursors that might give us an idea of how do these tumors actually arise, because until then we had really no good sense of this. And what originally started being reported as early as 2001 in the most famous papers by Pike et al. Here in the middle, they started observing the following types of things. A higher fraction of proliferating cells were found in dysplastic areas, and accumulation of puberty was observed in severely dysplastic lesions in the fallopian tubes, not in the ovaries. Following that initial study in 2001, a whole slew of other studies were published, illustrated here, and this is not intended to be completely comprehensive, just to illustrate the point, but what they also found was that when they did find occult cancers, they oftentimes involved the fallopian tube. And in one study out of the Brigham, what they found was fimbrial involvement of the fallopian tube. And so at the time, the interpretation of this was, okay, well, if you have a BRCA1 mutation, you're at risk of getting ovarian cancer, you're at risk of getting breast cancer, you're at risk of getting primary peritoneal cancer, which looks a lot like high-grade serous cancer, and you're at risk now of getting fallopian tube cancer. But it wasn't really until 2006 when Chris Crum at the Brigham started thinking about this and said, this is really weird. I mean, it's kind of paradoxical. We're looking for early ovarian cancer, but people keep stumbling on the fallopian tube. And back then, pathologists, I myself being a pathologist, we would routinely focus on the ovary when we were looking at these prophylactic specimens. We would cut the ovary and submit the entire thing for histological analysis. But the fallopian tube, which if you look at WHO statistics, exceedingly uncommon to find a cancer, a primary cancer of the tube, essentially they would take a section through the tube, look at it, no cancer, moving on and look at the ovary. No one really paid attention. And so Chris Crum said, I think we might be onto something here. Let's see if we can look at this more carefully. And so he developed what's called the CFIM protocol, which stands for sectioning and extensively examining the fimbriated end of the fallopian tube. So the whole purpose of this was to look at the tube in its entirety, which people hadn't systematically done. You amputate the fimbriated end of the tube, section the rest of it as illustrated down here, but you focus on the fimbriated end by cutting at multiple cross sections to expose as much surface area as possible and look at it under the microscope. And so what did we find at the time? Well, just to give you a sort of a brief intro into the histology of the fallopian tube, there are two major cell types in the fallopian tube. There are ciliated cells, which are illustrated by the small arrow here, and secretory cells, which is the large arrow. And if you look at normal fallopian tube from women who have no evidence of disease, no high risk factors, and you look for P53, the tumor suppressor that is very often mutated in this particular cancer, and you look by immunohistochemistry where you're looking for brown staining, you see that P53 is typically not expressed in normal tubes. If you look for evidence of DNA damage as measured by phosphorylation of a special histone called H2AX that gets phosphorylated at sites of double-strand breaks, you don't see any evidence of DNA damage either. And if you look at proliferation by looking at MIB1 or Ki67, you don't see much proliferation. In fact, normal fallopian tube has about a 1 to 2% proliferation rate in secretory cells. But in this population of patients, now remember, these are all women with BRCA mutations who are getting prophylactic surgery. What they noticed in up to a third of all these patients was what was ultimately termed a P53 signature, which is a stretch of benign appearing secretory cells. So now there's no ciliated cells, just secretory cells, very strongly expressing P53 protein in the nucleus. Evidence of DNA damage as illustrated by these intranuclear foci, these dots within the nucleus, but had not acquired a proliferative capacity. And in a minority of these cases, you would find these signatures in continuity with what looked like an intraepithelial carcinoma, has all the histological, cytological features of cancer, high NC ratio, proliferation, and so on, retained within the basement membrane, but also retained the P53 expression, these intranuclear foci of DNA damage, but had acquired a proliferative capacity. And the clinically evident cancer, all the way on the right here, shared all these properties, suggesting that maybe there was something going on in the tube that was then manifesting as sort of the advanced disease we were looking at, but this was all histologic. One of the things that did strike us, though, was that almost all of this was noted in the fimbriated end of the fallopian tube. Whenever we found these lesions, 95% of them were limited to the very distal end of the tube, right? The part of the tube that's in the closest proximity to the ovary. And so you can start sort of illustrating what we think might be going on, these sort of plica, these finger-like projections at the end of the fallopian tube in the fimbria. You have normal epithelium that transition into this P53 mutated state. An intraepithelial cancer can arise that can invade and disseminate to involve the peritoneal cavity. And that was all fine and good, but again, it was sort of histologically descriptive. So we decided to see if we can take it one step further, and this is back now in 2007, 2008, where we laser captured all these entities from a series of women. So women who had normal epithelium, the signature, the stick, and the tumor, and asked whether, in fact, when you have stabilized p53 protein like we see by immunohistochemistry, does that mean that there's an actual mutation in p53? And sure enough, it did. In cases where we saw these signatures, there was a p53 mutation. So there was a mutation in cells that looked benign, that did not proliferate, but had evidence of DNA damage, and had a mutation in p53. And in any one women's samples, you had the same mutation here, and in the in situ cancer and in the invasive cancer, suggesting that there was a genetic linkage between these three entities. We have subsequently done further studies, and we have a paper, one of my friends likes to kid around, it's not under rejection yet, where we've actually done whole exome sequencing of all these entities and try to pinpoint the evolution of this from a genomic perspective. But one of the things we went back to look at afterwards was, okay, is this unique to the BRCA population, or is this something that we see in women in general? And so depending on the literature you read, anywhere between 20 to almost 40% of all women, regardless of risk, so we're talking about BRCA and non-BRCA, right, will have a p53 signature in their fallopian tube. Now remember, we don't think that these are malignant precursors. They're not proliferative. They don't look malignant. They just have mutations in p53 and DNA damage. And they're probably altered in some way by some checkpoint that prevents them from progressing. But if you have a p53 mutation, you're much more, BRCA mutation, you're much more likely to traverse this state and enter into what we call the intraepithelial or tick stage, where you have an actual carcinoma that may be found in isolation before it invades, or in conjunction with an invasive cancer that involves the ovary or peritoneum. Now, again, in work that we've done back in 2007, and now we've done much more rigorously with whole exome and whole genome analyses, we know already that p53 mutations, not only at the morphologic level, but actually at the genetic level, is the first alteration. It's a truncal mutation in this cancer. And 100% of high-grade serous cancers have mutations in this gene. So it's an obligatory event, right? Unlike some other cancers where it's 50% and or occurs much later in the sequence, like in colon cancer, this seems to be the first event, an obligatory event. Studies from a number of other groups have actually started studying the intraepithelial carcinoma and have reported telomere shortening, which is a hallmark of precancerous lesions, and copy number alterations, even in this intraepithelial tick stage before becoming invasive. And so the question that has come up often after sort of these descriptive studies were published was, well, do all high-grade serous cancers come from the fallopian tube? And I think the short answer to that is we don't really know. The data does not say 100%, but these are two studies from probably two of the best pathology groups in the country, one at the Brigham and one at Hopkins, where they looked at this question, and they found that if you look at papillary serous carcinomas, you'll find that high-grade serous cancers, about two-thirds, 6 to 7% of them will have what we call a stick, a serous tubal intraepithelial cancer in their fallopian tubes in association with the cancer itself. And the other study showed about 61%. Depending on who you read these days, since these studies came out, it's anywhere from 50 to 80% have been associated with precancerous lesions in the fallopian tube. Well, that's high-grade serous cancer. What about primary peritoneal carcinomas, which I told you is a very similar type of cancer? And again, here, if you use the CFIM protocol, so if you extensively examine the fimbriated end, people are reporting about 47% of these cancers have a precancerous lesion in the fallopian tube, and this ranges to 45 and up to 55%. So perhaps even up to half of all primary peritoneal cancers have some origin or some genesis in the fallopian tube. When you look at the population of mutation carriers, so again, going back to women who have a BRCA mutation but don't have evidence of disease, how often do we find these precancerous lesions in isolation? So this is a collection of 3 studies that were published in the last few years. If you add it all up, you get about 1,000 cases and an incidence of about 4 to 5%. Some people think it's closer to 8% of mutation carriers will have an intraepithelial lesion in their fallopian tube at the time of prophylactic surgery. So again, back to our schematic of what we think is going on in high-grade serous cancer with the evolution from normal secretory cells through these pre-invasive precursor lesions to then invasive cancer that can exfoliate and involve the surface of the ovary or peritoneum. And this idea was an interesting one back in 2007, and I always like to quote Peter Medawar, who has said, "The human mind treats a new idea the same way the body treats a strange protein, and it rejects it," which is exactly what the journals did when we submitted our first set of papers, because they were like, "But this is ovarian cancer, what are you talking about?" So it took us a while to get that point across, but since that time, people have been using the CFIM protocol at sites throughout the US, Europe, China, Australia, and have seen the same thing. So people have appreciated the fact that a lot of high-grade serous cancers are coming from the fallopian tube and have identified these precancerous lesions. This is actually relevant because for decades people are looking for a consistently identifiable precursor in the ovary and weren't able to find it. So now we see that it's actually relatively easy to find precursors in the fallopian tube. And how come we can't account for all high-grade serous cancers coming from the tube? And this has been somewhat of a debate. A lot of explanations have been proposed. Sampling errors, meaning that if you don't section fully into the block, you can miss some lesions. We actually had this happen to us. We were collecting samples to look for a particular marker that I'll actually touch on briefly later. And I was looking for benign fallopian tubes. So I wanted just normal fallopian tube. I wanted to examine the state of expression of some of these proteins in normal. And so we had gotten a collection of 75 tubes from women who had either had breast cancer or had a family history, but their tubes were signed out by the pathologist as being morphologically benign. I was like, great. So we take these tubes, we section into them to do our study, and we came across intraepithelial cancers in 3 of them, which was not supposed to be there. We called the oncologist, we pulled back the old histology. The histology was benign. It's just that when you cut into the block, now you found a cancer. And when we went back, 2 of those patients actually recurred about 4 years later. So sampling, we've actually had the experience, can actually make a big difference. The more you sample, the more you're likely to find. That being said, though, even among GYN pathologists, there's a lot of inter-observer variability. So, the reproducibility of defining a stick lesion is not 100%. There's also the fact that based on the nature of the P53 mutation, sometimes you don't get stable mutant protein expressed, and so you have these P53 negative sticks that can make it more difficult to sort of call something a stick lesion. Lesions in transition between the P53 signature and the stick are difficult to diagnose. There are no defined criteria for that. And oftentimes the precursors can be consumed by the cancer itself, so there's no normal tissue to look at. So that all being said, I think there are other people who are proposing that there may actually still be other sources of ovarian cancer. The ovarian surface epithelium itself, the interperitoneal tubal junction has been described more recently, and then Alex Nikitin published a paper about the ovarian hilum, and the mouse has a niche of cells that may be precursors to this cancer as well. So I think that we can appreciate that the tube is contributing to the genesis of this disease, but it's not a completed story. I don't think we have all the answers yet. Now, at the same time that this is happening, this actually happened without us knowing, sort of in parallel. The Cancer Genome Atlas was in full swing. And the goal of the Cancer Genome Atlas was to provide a comprehensive sort of panoramic view of human cancer so we could really understand the genetics of this disease. And among the first 3 that were studied were brain tumors, lung cancers, and ovarian cancers, because we don't have early detection for any of them, and our therapeutics for them are relatively limited. And what you find when you look at GBM, you find that by mutations, there's a nice spectrum of alterations in again, all your favorite cancer genes, P53, P10, NF1, etc. Same for lung cancer. In fact, same for breast cancer. But when you look at ovarian cancer, I remember being at the Broad when they released this data, and what stuck out to everyone was the fact that you see this almost obligatory, as I mentioned, mutation in P53, but hardly any other recurrent somatic alterations, there was no other cancer that had been described like that. This is very strange. But when you do a pan-cancer analysis, so now you're looking at tumors from left to right, breast cancers, bladder cancers, colorectal, GBM, head and neck, etc., ovaries here, and you look at copy number from chromosome 1 all the way down to 22, what you begin to appreciate is that ovarian cancer is the most complex cancer in terms of genomic terrain that's ever been described. There are more amplifications and deletions in ovarian cancer than in any other cancer described so far by the Cancer Genome Atlas. And this is contrasted by the bottom panel, which is actually looking at somatic mutations. And you can see that even though you have all these copy number alterations in ovarian cancer, it's actually lacking very many recurrent somatic alterations. So the disease is defined by copy number alterations, not by mutations. And so the convergence of these two approaches, meaning studying the histological pathogenesis of this cancer and the genetics, have led us to sort of appreciate that high-grade serous cancers can, a majority but not all, have an origin in the fallopian tube that progress through these precursors we call the PP3 signature and the tubal intraepithelial cancer. They have the hallmarks of having DNA damage early on with mutations in P53 that are retained throughout the transformation process. And as people are doing more work, we're starting to appreciate that there's telomere shortening in these precursors and the beginning evidence of copy number alterations before the clinically evident cancer displays all the same features. So take-home messages. Precursors to high-grade serous exist in the tube. The CFIM protocol is a really important clinical tool for assessing risk reduction samples, and now people are actually deploying this to look at women at elevated risks, meaning not BRCA1 or 2 mutations, but a strong family history without a defined genetic alteration. They're characterized by early mutations in P53 and widespread copy number changes, and it seems that the stick lesion is a precursor for this disease. In work I really didn't touch on very much, and this is again work that we're doing in collaboration with Victor Velculescu, where we've genomically analyzed all these precursors at the whole exome and whole genome levels, suggests that the tube is the precursor, the site of origin for a majority of these cancers, and can directly give rise to metastases while bypassing the ovary itself in certain cases. So that's all fine and dandy. We're still working on that, so are a lot of other people, and the more you do, the more questions you actually have. What are the implications of this for early detection? There's been a tremendous effort. Many of you are familiar with the British study that was published at the end of December, the UK CTOC study looking at early detection, trying to see if there's a signal there, with a lot of focus on the ovary as the site of origin, the source for biomarkers. What about prophylactic surgery? We take out the tubes and ovaries as women are put into a surgical menopause. Can we afford to think about a two-step reduction where you take out the tubes first and maybe take out the ovaries once the women have entered their own menopause? Is that as effective? Is that as safe? And then therapies. And I won't stand here and tell you I'm going to give you the answers to all these, because I think these are things that are still being discussed and debated and researched in the literature, but I will give you an idea of how we've come attacking this question, and that's because from the research bench we've tried to utilize this new insight to model this disease in ways that we didn't have abilities to do before. But before I talk about the models themselves, I want to talk about a particular protein that actually led us to do a lot of the work that you're going to hear about, and that's PAX8. So PAX8 is a homologue of the Drosophila paired gene. So paired was cloned in 1986, and shortly after it was cloned, it became apparent that it's actually a member of a larger network of regulatory genes that control segmentation, patterning, and organ development in the fruit fly. It is evolutionarily highly conserved. We see PAX genes from nematodes all the way to humans. And in humans, we know that in primates, we know that the PAX8 is involved in Mullerian development. There's a study that was done about 7 years ago where they knocked out PAX8 in a mouse. And if you knock out PAX8 in a mouse, you don't form a female reproductive system. There's atresia of the entire reproductive tract. So no uterus, tubes are atretic or hydrosalpinx. The ovaries are perfectly fine. So PAX8 is actually essential for the development of the Mullerian system. In studies that we did with Bill Hahn at the Broad, we found that PAX8 is the most differentially expressed gene between ovarian cancer and any other kind of cancer. It's retained in the adult secretory cell, and this is an immunohistochemical stain where you're looking at human fallopian tube, and you can see nuclear expression of PAX8 in secretory cells, not in ciliated cells. If you look at tumors, nearly 100% of all high-grade serous cancers retain expression of PAX8, and if you knock down PAX8 in ovarian cancer cells, it oftentimes leads to apoptosis or senescence in many, but not all, cancer cells. And so this has sort of led us to think about PAX8 as a lineage-specific transcription factor, and one of the things I was intrigued by for a long time was trying to understand, well, how does PAX8 do all this, right? How does it govern development of the female reproductive tract? What is it doing in cancer? Is it doing the same thing in cancer that it's doing during development? And so to begin to answer this question, we took advantage of the fact that we had previously characterized some ovarian cancer cell lines that were deemed by prior studies to be more representative of high-grade serous cancer, the Osaho, the Kormachi, and the JHOS4 cells, and compared those cancer cells to 3 fallopian tube cell lines we had developed in the lab that were immortalized but not transformed, so sort of normal in cancer. And we used a combination of RNAi knockdown for PAX8, RNA-seq, and then ChIP-seq to really try to understand what is the transcriptome and systrome that PAX8 governs, and is it different in the benign versus malignant state. So what you're looking at in panel A here is just simply a Western blot illustrating the 3 different siRNAs that we use for PAX8 in all the 6 cell lines. So you see efficient knockdown by our RNAi reagents. And then on panel B here, what you're looking at is unsupervised clustering after PAX8 knockdown in all these cell lines. And what we found was that they clustered into 2 groups, the benign and the malignant. The benign clustered together and the malignant clustered together. But one of the things we noted was that in looking at the changes in genes that were affected by PAX8 loss, in the benign group, there are hardly any genes that actually were affected by PAX8 knockdown, which surprised us. But there are many genes, tens to hundreds of genes affected by PAX8 knockdown in cancer cells. So we thought, well, is this because maybe other PAX genes are redundant with PAX8 in secretory cells? But if you look at RNA-seq data at all the other PAX genes in these cells, really PAX8 is the only one that's expressed in the adult secretory cell on the bottom here. So that really couldn't explain it. So we wondered whether, in fact, what is happening is that there's remodeling around, an epigenetic remodeling around the PAX8 binding sites between the normal and malignant state. And so what you're looking at here is a genome browser view of a highly conserved PAX8 peak on chromosome 8 that we saw both in the fallopian tube and in the high-grade serous cancer samples. And when we collected all our peaks to define a PAX8 binding site, because at this point there was no clear defined published PAX8 binding site, when we aligned all our peaks, the binding site that most closely resembled what came out of this was a PAX2 binding site, suggesting that in fact we were pulling down a PAX homolog out of these Müllerian cells. So then we looked at, well, what's the status of the systrome, the binding sites? Where's PAX8 binding in the genome? And found again that if you look at unsupervised pairwise clustering, we see that the tubes cluster together. And I'll just pause for a second just to let you know that these 3 fallopian tubes are derived from 3 different women, and they were immortalized in 3 different ways. And we did that on purpose because we didn't want this to be a reflection of a particular immortalization protocol or a particular tube that we got from Mrs. Smith. This was intended to look at what's actually the underlying biology as opposed to our methodology of generating these lines. And you can see that they tend to cluster together, whereas the tumor cells tend to actually have unique peaks that are not present in the normal. So it says that it appears that they're actually shifting from where they are in the normal state to different parts of the genome in the cancer state. And when you look at this by k-means clustering, you can see that there are 4 major clusters. There's a cluster of peaks that are common to all cells, all Müllerian cells, both normal and malignant. There are a cluster of peaks that seem to be more common in the benign cells and not so much in the cancer cells. There's an acquisition of peaks that we see only in the cancer cells and not in the normal cells, suggesting in fact there is, again, a shifting of the PAX8 systrome between the benign and malignant cells. When we looked at where is PAX8 binding in the genome, we noticed that actually less than, I think, 7 or 8% of PAX8 peaks were present near promoters. The vast majority were intronic or intragenic sequences. And now we've added onto this an analysis looking for enhancer regions and super enhancers, and it seems that PAX8 is binding not infrequently near enhancers and actually some super enhancers as well. So it actually is governing the genome in ways that are not sort of canonical with binding a promoter and turning on a particular gene. It actually may be doing much more of an epigenetic modification. And if you look at the genes that are affected and the sort of pathways that are affected by PAX8, you notice that a lot of the things that are affected are proliferation, blood vessel development, vascular development, adhesion molecules, morphogenesis of blood vessels, things that are needed during development and things that cancers co-opt during malignancy, but not so necessarily important during the adult mature sort of differentiated state. And we wonder whether this may have something to do with why we don't see an effect on the normal benign cells when you knock down PAX8, but it's much more pronounced in the malignant state and when you knock it out in an animal during development. So the next question that we ask is, well, is PAX8, is this sort of rewiring of the PAX8 systrome in cancer have anything to do with the partners that it may be working with. And one of the things we're working on now is trying to define the Paxate protein complex to figure out who does it work with to do what it does. But another way to ask this question is the Paxate binding site in the vicinity of other known transcription factors, and is there an enhancement for this at all? And when we did this analysis of all 6 cell lines, both benign and malignant, what we found that is in the malignant cells, there seemed to be an enrichment of TAD binding sites near the Paxate peak, near the Paxate site. And so when I got this data, I was like, what's TAD? So TAD turns out to be part of the hippo-YAP pathway, which is an oncogenic pathway that is driven by YAP binding to TAD and driving expression of a lot of genes that govern proliferation, survival, transformation. So we were intrigued by this and wanted to see if in fact this could be part of the reason why we see reprogramming in the system. So the first thing we did was, again, by RNA-seq, look to see whether all the TAD proteins are present in the samples, and you can see that in fact to varying degrees they all are. We did immunofluorescence to show that Paxate, TAD using a pan-TAD antibody, YAP, which is the actual transcriptional activator, and P300, which is a known co-activator, are all expressed in the cell lines that we were interested in. So then the question becomes, well, do they actually interact? Are they actually functioning together? And to do this, we used the proximity ligation assay. So for those who aren't familiar with that assay, it's actually a very neat assay. If you suspect that 2 proteins interact, like you look here on the left, right, this blob A and blob B, what you can do to ask whether in fact you can detect that interaction is use an antibody against protein A and protein B. And then with secondary antibodies that are unique to each of these, which are species different, so you have to use a mouse and a rabbit, or a rabbit and a chicken, that have primers linked to them. If the proteins are close enough together, these primers can actually start a PCR reaction and generate a product that then is recognized and can become fluorescent. So the only time you get fluorescence, or you get a product from the PCR reaction, is if these 2 proteins are close enough together. Otherwise, right, they can't form the PCR primers can't come together to generate this substrate. And so what you can do is you can knock out one gene and see if in fact the signal goes away as you would expect, and you can look at a variety of different aspects. But we use the proximity ligation assay to ask whether we could see Paxate interacting with TAD proteins. And the advantage of this assay, and you can see down here that you can do both fluorescence and in situ immunohistochemistry, is that not only does it tell you if 2 things interact, it tells you where they interact, right? So it's not just like a Western blot co-IP which says 2 things interact. This will actually tell you where in the cell, which compartment in the cell do the interactions occur. And so when we looked at whether in fact we could see interaction between Paxate and the TAD proteins, what you find is that, and hopefully you can see it with the light here, but what you can find here is that there's red dots within the nucleus of these cells, suggesting that in fact these 2 proteins are actually interacting together. Paxate was also found to interact with YAP, which is the activator, the oncogenic factor, and with its known coactivator protein P300. But if you do RNAi for Paxate, the signal goes away, and if you use no primary antibodies, there's no signal, suggesting that these interactions that we were seeing are actually real. And we're now following up on this to see if in fact this interaction is necessary for YAP-mediated transformation, which has been reported in this setting. So Paxate is an interesting protein. There's a lot of neat work done on it. And based on some of the work I already told you, we decided that this was a good candidate to think about generating a mouse model for high-grade serous cancer. And so in order to do that, and this is sort of the systems of the animal systems that we've developed, both genetically engineered mouse models and PDX, or living tissue bank models, as well as cell line models, immortalized secretory cells, which I mentioned to you already, and then primary cells that enable us to study the susceptibility of the fallopian tube to transformation. So the genetically engineered model was really, again, based on the fact that Paxate is expressed in the adult secretory cell in almost 100% of these high-grade serous cancers, and fortuitously the antibody works in the mouse, and you can see expression in the fallopian tube, not on the surface of the ovary, and at higher power you see that same mosaic pattern of expression in secretory cells, but not in ciliated cells. And a talented postdoc in the lab, Ruth Parrots, who now has her own lab in Israel, decided to see if she can take advantage of this, so she took a Paxate RTTA mouse and crossed it to TETO-CRE, which essentially enables you to induce with doxycycline the expression of the CRE recombinase from the Paxate promoter. We crossed that to a LAC-Z ROSA26 system to see if we could quantify exactly where is Paxate expressed in the mouse. And so for those who are not familiar with the anatomy of the mouse, It's different than humans. The mice are a little bit different. They have two horns to the uterus, so it's a bicornuate uterus. There's not a single cavity. The fallopian tubes are a tiny coiled structure at the very tips, and the ovaries next to it. And in the mouse, they're actually enveloped by what's called the ovarian bursa. There's a sac that exists in the mouse that doesn't exist in humans. So when we looked in our animals, in the absence or presence of doxycycline, you see that there is blue staining, beta-galactosidase staining in the fallopian tube, which at higher power becomes very readily evident that the tube is targeted by the system. We looked at other organs in the body to see where else this was expressed. The only other tissues that express PAX8 are the renal system, renal collecting proximal cells. We worried about that as a possible a cancer that we weren't interested in. And I'll show you that unfortunately for us did not create a problem. So we thought we had a good system where we can induce PAX8-Cre from the PAX8 promoter. And then the question is, well, so what do you model, right? So at that point, we went back to the Cancer Genome Atlas. And one of the things that came out of that study was that the description that about half of all high-grade serous cancers have a defect in genes involved in homologous recombination, including germline and somatic alterations in the BRCA genes. And as I mentioned, p53 is sort of an obligatory event. So looking at HR dysfunctional tumors, we thought this would be a good system to try to model. Sort of the other half of the pie, which I'll try to touch on it towards the end, has alterations that seem to be mutually exclusive with BRCA alterations, and we've tried to model that as well. But we started with the BRCA. And so we collaborated with my colleague Danielle Denalesco at the Brigham, who is a professional mouse modeler. I'm not. And she had a lot of these alleles already, so we looked at BRCA1 and BRCA2, homozygous deletion versus heterozygous. We looked at p53 mutations versus deletions, and we looked at the contribution of PTEN, because if you look at the PI3 kinase PTEN pathway, it's about 77% of high-grade serous cancers have some dysfunction in that pathway. And we asked, if you generate these double or triple knockout animals and cross them to PAX8 and induce CRE, what happens? And so what we found, again, harkening back to that bursa that I told you exists in the mouse, we found that after doxycycline induction, we found that these bursas become cystic. About, depending on the genotype, about 6 to 8 weeks after induction, we saw these cystic lesions in the fallopian tube. If you look under the microscope, what you're looking at here is a normal fallopian tube on the left, and this is low power. And then after induction, what we found is that normal architecture, the size was actually retained, but the epithelium had been completely replaced by what looked like intraepithelial cancer. And what you're looking below that is a higher power version of that. So this was sort of the equivalent of a stick lesion, the intraepithelial cancer I described in humans. And these lesions would progress to advanced disease that would involve the entire peritoneal cavity. We didn't see a lot of ascites in our model systems, but we did see some, but we saw a lot of bulky disease in the abdomen that affected all organs that were examined at necropsy. So it seems that induction through PAX8 in the tube can cause precancerous lesions that evolve into cancerous lesions. And we stained for all the typical cytokeratid markers. But one of the things that we were interested in asking was whether we had actually been able to recapitulate the disease in a way that had nothing to do with the genes we altered. So let me tell you what I mean by that. A few years ago, we looked at a particular protein, STADMIN1. So STADMIN was described by Ramon Parsons as a tissue biomarker for PI3 kinase pathway activation. And it's a protein involved in microtubule dynamics. And we thought to look in these precursors in the fallopian tube to see whether STADMIN is a marker of early cancers. And what you can look at here, here's normal, here's p53 signature, and here is the stick lesion. And you can see that there's a very strong induction of STADMIN in these in situ cancers that you don't see in normal or even the p53 signature. And these seems to correlate with loss of p27. And this was in human. So we thought, okay, let's see if our mouse recapitulated this. So what you're looking at the bottom here is normal fallopian tube. You see PAX8 expression in the bottom left here, as expected in the secretory cells. These are not proliferative. They don't express STADMIN, and they have p27. But in the precursor lesions, we see that you get increase in proliferation, acquisition of STADMIN expression with loss of p27. Suggesting that even though we'd only manipulated 2 or 3 genes in our system, we'd been able to recapitulate a phenomenon that we'd seen in humans before that was independent of the genes we affected. But most importantly, we wanted to know if we retained the clinical and genetic hallmarks of this cancer. So one of the things we looked at was whether these cancers elaborate CA125. And when you look at tumor-bearing animals in the periphery, we developed a CA125 specific for mouse CA125. You can see the tumor-bearing animals had more CA125 in the periphery than their control litter mates, that this CA125 was actually coming from the tumors, because you can look at 4 different tumors here compared to normal, and you can see that the tumors express CA125 and the normal does not. The other hallmark, as I told you, is DNA damage, and when you look at these tumors, you see that there's evidence of H2AX phosphorylation. Again, this histone that gets phosphorylated at sites of double serine breaks, we see that evident at the level of the Western blot in these cancers, but we wanted to see whether at the level of the genome, had we recapitulated some of the complexity that seems to be the hallmark of the disease. And so we used array-CGH, comparative genomic hybridization, to look at some of these tumors. And here's an example of 3 tumors that we studied, and it was sort of reassuring to find that even though, again, we'd only manipulated PP3, BRCA1, and or P10, depending on the genotype, we had actually been able to recapitulate the complexity of amplifications and deletions that are the hallmark of this cancer. So for instance, MYC, which is the most commonly amplified gene in high-grade serous cancer, is amplified in these tumors. KRAS, which is not mutated in high-grade serous cancers, but is amplified in these models. IMPP4B, which is a relatively new tumor suppressor, is also deleted in these models. And so again, we've been able to recapitulate the complexity of this cancer by targeting the right cell with the right combination of genes to create a model that seems to really retain the genetic hallmarks of the disease. So then we did a few experiments that have left us scratching our head a little bit, but we're looking at it a little bit more carefully. So if you do salpingectomy in these animals, you find that, as one might expect, since you're targeting the tube, then you would see no transformation, no development of the stick lesion, and no cancer anywhere else. So that made sense. If you do hysterectomy, you don't block the development of the precursors in the fallopian tubes or the dissemination to the ovary or peritoneum. But if you do oophorectomy, which we're not targeting, right? So you're not targeting the ovary, but if you do oophorectomy, you don't block transformation in the tube, but you seem to attenuate greatly the ability of the tumors to then leave and go to other parts of the peritoneal cavity. And this was a paper, observation we've reproduced, and more importantly, Ron Buchanovich and Lan Coleman from University of Michigan just had a paper last month where, using a different system, they argue for the same effect, which is that the ovary, even though it may not be the site of origin, actually seems to play a major role in the ability of these cancers to disseminate and seed the peritoneal cavity. And whether this is seed in soil or some other factors in the ovary that attract these cells there and enable them to grow before they go elsewhere remains to be addressed, but it's an interesting observation. So we've gone back and looked at this sort of the genetic pie chart of high-grade serous cancers, which has now been sort of elaborated by some of the work from David Botel, and as again, developed the BRCA2, BRCA1 models. We've now looked at some of the other HR genes like PALB2, which is the closest thing to BRCA3, to see whether PALB2 not only substitutes for the BRCA genes in transformation, but do other HR genes predict response to PARP therapy, for instance. And then the other side of the pie, the cyclin E, which I'll talk about in a minute, cyclin E, we're looking at that as well, and we've developed a cyclin E mouse. So this sort of represents a bit of our approach to modeling of this disease from the fallopian tube. PDX models, which I won't talk about, are one way to do it. Genetically engineered models are a different way to do it. So in summary, we can target the tube in vivo. And if you target it with disease-relevant alterations, you can recapitulate the pre-invasive and invasive morphological hallmarks of the cancer. The stick lesion does appear to be the precursor of high-grade serous cancers, and importantly, these mouse tumors retain both the tumor and serological biomarkers that define the human disease. A lot of genomic instability in these tumors, as we've shown. Salpingectomy, as expected, blocks development of the cancer, while oophorectomy doesn't block it, but seems to contribute to its ability to disseminate. So I'm going to finish with a little bit on the genetic landscape of this cancer and looking at another side of the pie, cyclin E. So cyclin E was described as being amplified in about 20% of high-grade serous cancers. The interesting thing that came out of the TCGA was that they reported that cyclin E was mutually exclusive with BRCA mutations. So you never see the two together. And at the time, no one really understood why that was. We decided to look at this more carefully because cyclin E, as many of you know, is a partner with CDK2, which is a druggable target. So I'll mention that as well. So cyclin E exists and is encoded on chromosome 19q12. It's one of the 3 or 4 most common amplicons in high-grade serous cancer. What does cyclin E do? Well, we know that cyclin E partners with CDK2 to govern transition from G1 to S. And if you have too much cyclin E, either through amplifications or overexpression, we get what we call unscheduled entry into S phase. So G1 gets shorter and you enter S phase before the cell is ready. You get misfiring of origins, and that creates a whole host of problems that have been described. You get what's called replicative stress as a result, and centrosome amplification. So we decided to look and see whether we could understand the timing of cyclin E deregulation in these early precursors. So one of the advantages of being able to study and look at precursors, now that we know where to look, is that you can start asking the question, well, when does any given alteration occur? Is it an early event? Is it a late event? We would argue that early events may be involved in transformation in neoplastic events, as opposed to late events, which could be a consequence of genomic instability. So what we did was we used a clinically annotated tissue microarray, and this is work that was done by Alison Karst, where we did FISH for cyclin E and for centromeric control. And long story short, what we basically found was that about a quarter of these had amplifications in cyclin E. If we then looked at the protein level in the same tissue microarray, again, you can see a correlation with high levels of protein in about a quarter of these cases, and it tend to correlate with copy number. So then we said, okay, good. So now we have the tools to look at this in early cancer, and so we decided, let's go back to our idea of how these tumors arise and ask, where do we see cyclin E deregulation? And so here we're looking at an example where you have normal fallopian tube epithelium, stains for P53 up to here, right? And this is not proliferative at this point here. So this is consistent with a signature lesion, right? P53 stabilized, not proliferative, and yet we see expression of cyclin E as early as the P53 signature. And that was about a third of signatures could see that. In the stick lesions, which are now in intraepithelial cancer, we see that about 2/3 of cases had concurrent cyclin E expression. And in a paper we just submitted with Yimeng Shi, we actually went back and redid this using FISH to actually look at copy number, because this is obviously looking at protein, and we see the same thing. So cyclin E deregulation at the genetic level can occur early in this cancer. And so then we thought, okay, maybe we could model what's going on by going back to one of our model systems, which is the immortalized model system, where we can take secretory cells, immortalize them, and use them as an in vitro system to look at the contribution of any genetic alteration. And so again, Alison obtained fallopian tube cells, isolated them, immortalized them with telomerase and a mutation in P53. Again, we argue that every model that we make should have a mutation in P53 because it seems to be an obligatory event in this cancer, and simply ask what happens when you overexpress cyclin E versus a vector control. And so what you're looking at in panel A here is sort of the expected stuff. You get activation of CDK2 with higher levels of cyclin E. You retain your Mullerian and epithelial markers, but you get an increase in proliferation that you see in panel B, and some clonogenic growth, but nothing dramatic. You lose contact inhibition, which had been reported by Steve Reed before, and you get a modicum of anchorage-independent ability by these cells, but they're clearly not completely transformed, and I'll come back to that. One thing we did notice, though, is that cyclin E was causing increase in DNA damage, and this was measured by immunofluorescence for that histone, that H2AX that gets phosphorylated at sites of DNA damage, and you can see higher levels of H2AX in cyclin E expressing cells. We confirmed this with a COMET assay where we saw longer tails in the cyclin E cells compared to its vector control. So there's stress, there's damage that's being induced by cyclin E. And importantly, when we profiled these cells, we initially did this with a PCR array, now we've done it with RNA-seq. One of the things that jumped out to us was the fact that the genes, and this is a volcano plot, so the top right are the genes that are statistically upregulated in cyclin E expressing cells versus the top left, which are downregulated. And the things that jump out are the fact that the proteins, the genes that are upregulated are ones involved in BRCA1, Fanconi anemia, homologous recombination, replication fork protection, intrastain cross-link repair. When we looked and confirmed this by Western blot, in fact, you can see higher levels of FANCD2, XRCC2, which was one of the RAD51 paralogs, and a host of other genes as predicted by this expression data. If you look at the TCGA, you find that, in fact, by TCGA, we also see an enrichment in genes in the Fanconi and HR pathway in cyclin E amplified tumors. Now, this is where it got really interesting because David Botel, who's a good friend, he works at the Peter Mac in Melbourne, Australia, had been working on a synthetic lethal analysis of cyclically amplified cells. So what he did was he said, okay, let's look at this cell line encyclopedia. Let's take out all the cells that have cyclically amplifications versus the ones that don't, and do a genome-wide RNAi screen to find what is synthetic lethal with cyclically versus non-cyclically amplified cells. And what he found was that the genes that are synthetic lethal are the exact same genes that we found are upregulated when you force cells into S phase with cyclin E. So, whoops, let me go back. So FANK D2, XRCC2, CDC25, BRCA1. So now we had an explanation for why BRCA1 and cyclin E are mutually exclusive, because they're synthetic lethal. So if you think about it, we're starting to come up with the idea that cyclin E stresses the cell, it pushes the cell into S phase, it causes DNA damage. In order to tolerate that, you have to upregulate your DNA repair pathway, right, so that you protect those replication forks. And if you don't, you're going to die. So you need BRCA1. So this is the opposite way of thinking. This is not BRCA1 is mutated. This is actually BRCA1 is necessary, right? Now, why is cyclin E interesting? Well, as I mentioned, cyclin E partners with CDK2. CDK2 is an enzyme, right? And it's that partnership that drives the G1S transition. Now, if you look at the sensitivity and dependency of cell lines that have amplification in cyclin E versus those that don't, they're exquisitely sensitive to knockdown of cyclin E or CDK2. So these cells need cyclin E and CDK2. They become addicted to this protein complex, whereas these other cells really don't care. And that makes it obviously a potential for using cyclin E as a biomarker for CDK2 inhibition. And there are a lot of CDK inhibitors in development. We've all heard of the palbociclib's success in breast cancer for CDK4/6 inhibition. CDK1/2, however, there are inhibitors. Unfortunately, none of them are really specific to CDK1 or 2. They always tend to hit CDK9 and 7, and you get a lot of toxicities as a result. But dinaciclib is one that Merck made that was interesting to us in terms of using it as a proof of principle. And one of the things we noticed with some of these drugs is that, as expected, cell lines that had amplifications, like OVCAR3, are much more sensitive than cell lines that don't to these drugs that target CDK2. So one of the things that we wanted to understand further was, well, so the druggability of CDK2, and for those who are familiar with CDK2 inhibitors in the clinic, they've always been sort of thought as, not too exciting. Part of it may be because we haven't had a biomarker to select out the tumors that are actually responsive for it. But what we noticed was that the drugs are never as effective as knocking down cyclin E or CDK2. And so we wanted to go back and look at our original cell line model system to ask, is something else cooperating with cyclin E to fully transform these cells? And is that an opportunity to intervene? And so Allison went back to her cell lines and said, okay, well, one way we could do this is do a gain-of-function screen and use a whole genome approach. But the other way to do it is say, well, we know that ovarian cancer is kind of weird, right? It's got all these amplicons. And there are 63 recurrent amplifications in ovarian cancer. So all those peaks, the red peaks. So we actually made a library of all the open reading frames of all the genes in the 63 recurrent amplicons and used that as our gain-of-function screen instead of a whole genome library. And this is sort of the layout, right? We have the library here. We have our fallopian tube cells that have mutant p53 and cyclin E. And we simply asked what happens when you overexpress these different genes and looked at anchorage-independent growth, and ultimately we did it in vivo as well. And so we looked at ultra-low attachment plates and soft agar formation using the screen, and as you might imagine, you're going to get hits. There were some common hits between the two, but I'm going to draw your attention to two things that jumped out. One was AKT1 and AKT3. Turns out AKT2 was not in the library. But one of the things we noticed was that if you look at, so one way to do it is a gain-of-function screen, which is what we did. The other thing that David Botel said is that, okay, well, what if we take dinaciclib, the one drug that seems to be okay but has some toxicity, and we ratchet down the dose so that it's less toxic, and we come in with a small molecule library to see what would cooperate with it, right? So that's what they did here. And this is sort of the layout of their screen. This is a paper that's under review right now. Long story short, what came out of that was a AKT inhibitor, right? In fact, an AKT2 inhibitor. So MK2206 showed that in amplified cells versus non-amplified cells, there was tremendous synergy between dinaciclib, a CDK2 inhibitor, and an AKT inhibitor, right? And what we found really interesting was that I mentioned AKT2 was not in the screen. so I didn't pop out. Any one of the AKTs can cooperate with cyclin E to transform these cells. So why AKT2 as a drug? It turns out AKT2 is right next to cyclin E in the amplicon, so that when cyclin E gets amplified, it takes AKT2 with it, and that way cooperates to transform these cells. So that's a possibility that now we're pursuing to see whether, in fact, we can take a drug that's suboptimal in the clinic because of toxicity, reduce the dosage of it, and look at combination with an AKT inhibitor to see if we can see some efficacy. And in studies that are published by the Sanger, you can see that in cyclin E amplified cells, the most effective drugs tend to be within the AKT inhibitor family. If we look at this in an animal now, so we look at dinucyclib and the inhibitor, you look at a line that's amplified, so OVCAR3 versus the one on the bottom, COV3 here, you see that the combination is able to either stunt growth of the tumor or actually cause some regressions, whereas the non-amplified cells don't seem to really care. So the combination seems to actually work in the cells that have this dependency. And so again, we're sort of working to generate this mouse model now to see if we can look at the pathogenesis of cyclin E-driven tumors in the mouse and see if, in fact, we can recapitulate this AKT independence. And we combine this again with a combination of looking at PDX models that have cyclin E amplifications with our GEM models to get a better sense of what's going on. And why is cyclin E interesting? And I'll end with this. If you look at the Cancer Genome Atlas, ovarian cancer has, like I said, about 20% of the tumors have amplifications in cyclin E, but for sure it's not the only tumor type that has amplifications in cyclin E. And in fact, uterine carcinosarcomas are approaching 50% with the current data in the TCGA. So we suspect that things that we learned from the study of what we've been doing in ovary may actually be translatable to some other tumor types in terms of dependencies and vulnerabilities that we could take advantage of in the clinic. And I'll end with that. I'll just thank some of the folks. These are some of the new people on the top here in the lab at Penn, with some of the old folks, not old, but older folks that were in my lab at the Dana-Farber. My new colleagues at Penn, which has been a real pleasure to work with. And my old colleagues, especially Chris Crum at the Brigham, who a lot of the work on the tube really wouldn't have happened without him. Michelle Hirsch and Daniela, who really helped spearhead a lot of the mouse model development, and colleagues throughout the US and the world that have been fun to work with over the years. Thank you.